So this video is going to be an overview of the 4 New York State labs used in the Regents exam. The first state lab is diffusion through a membrane. It's predicated on the fact that some substances are larger than others. Membranes are selectively permeable and will prevent certain substances from passing through because they are too large. Diffusion is the movement from an area of higher concentration to an area of lower concentration. Movement from lower concentration to higher concentration is called active transport and requires energy. This lab uses glucose, which is a simple sugar molecule, and it also uses starch, which is a complex molecule made of multiple glucose molecules. This lab also uses 2 indicator solutions. Lugol's iodine, which is the indicator solution for starch. It turns dark blue black in the presence of starch, and it starts amber colored. Benedict's solution is the indicator for glucose. It starts out blue, it turns orange in the presence of heat and glucose. You start the lab with an artificial cell with glucose and starch inside, and on the outside you have iodine. The cell is made from dialysis tubing, which is a porous material. After time passes, the iodine diffuses into the cell, reacting with the starch and turning the inside black. However, as you can see in the picture, the outside does not change color. Starch is too large to diffuse, and that's why the outside of the cell does not turn black. When you test the liquid from outside the cell, it tests positive for glucose. So the initial state is inside has glucose and starch, outside has iodine. The final state, inside the cell, has glucose, starch, and iodine, and the outside has iodine and glucose. The glucose and iodine will move from higher concentration to lower concentration until an equilibrium is reached. Once the equilibrium is reached, they will move back and forth, keeping a balance. This list on the left side is important to know, as the Regents will use a diagram to show what the initial state and the final state was. The next part of this lab deals with onion skin. When salt water is added to the onion skin, water will rush out of the cell membrane, causing the cell to shrivel up. The cell wall will remain the same size. The water concentration inside the cell is higher than that of the salt water, so the water will rush out in an effort to reach an equilibrium. This process is called plasmolysis. When you add distilled water, which is salt-free water, to the cell, the water will rush back in, refilling the cell. You can see in the picture on the left, using dye, what the cell looked like initially, then the shriveled up cell membrane state in the middle, and on the right after distilled water was added. The second New York State lab deals with beaks of finches. Finches have different sized beaks, and they have different shaped beaks, and it allows them to occupy different niches. The size and shape of their beaks will allow them to feed better in different environments. This picture on the left shows the variation in beaks of Galapagos finches. This diagram traditionally shows up on the Regents and will ask you to find which finch would occupy which niche based on the food type, the type of bill, or the size of the bill. This lab uses 3 different beaks: pliers, tweezers, and clothespins. There are also smaller seeds, seen on the left, and larger seeds used on the right. These are used to represent different food sources. Different beaks will have different characteristics, making them better suited to different environments. They will feed on different food sources in order to survive. The third New York State lab deals with heart rate. Your heart pumps blood around your body through your circulatory system. The blood carries oxygen to your muscle cells using red blood cells. When you're exercising, your heart rate will increase to help meet the oxygen demands of your muscle cells. You can detect this through your pulse rate. The harder you work, the higher the demand for oxygen, the faster your heart will beat. Squeezing the clothespin. When you squeeze the clothespin, it will fatigue your muscles, making it harder to repeat the performance the second time. Some people, however, will be warmed up and not only repeat the performance, but they can surpass it. This all depends on your particular skill set. If you text or play video games a lot, your fingers might be stronger and better able to squeeze the clothespin. The last New York State lab deals with relationships and biodiversity. This lab is an investigation to find which species X, Y, or Z, is most closely related to the fictional species Botanicurus, which has cancer curative properties in the scenario presented. The first part deals with physical characteristics. You look at cards that have the plant flower and the seeds on them. You can see the flower on the lower left-hand side and the seeds on the right-hand side. The next thing you will do is you will look at microscopic cross-sections of the stem. The cross-sections can be seen on the bottom right. You look at the plant cards and the slide to determine, based on looks alone, which plant is most closely related to Botanicurus. The second part of the lab deals with several tests for molecular evidence. You start with genetic relationships. Remember, the central dogma of biology is DNA codes for RNA, codes for proteins. All right? A chain of amino acids is a protein. You are asked to find the DNA pairing. Remember, A pairs with T, G pairs with C. Then, based off that DNA, you are asked to find what the mRNA would be. A pairs with U, G pairs with C. And then, based off that mRNA, you use the codon chart on the right to determine what the order of the amino acids are. 20 amino acids are coded for by 64 different 3-base RNA sequences called codons. Those codons and their corresponding amino acid are listed on the right. The second test is the test for enzyme M. Enzymes are proteins that speed up the rate of reactions. What you do is you put the chemical in the testing well and add a test solution to it. Botanicurus, Y and Z are all positive, X is negative. Essentially, the reaction is a baking soda and vinegar reaction. So X does not have vinegar in it, so it does not react with the baking soda and bubble up. Genetic relationship 3 is chromatography. Chromatography is separating pigments based on size. Smaller pigments travel up the paper further. Larger pigments do not. You place a dot of the pigment on the chromatography paper. You put the end of the paper in a developer solution and make sure not to let the dot be submerged under the liquid, and you wait for the pigments to separate by the water being drawn up the paper. Here's a picture of chromatography occurring and a separation. And on the right is what the chromatography would look like for Botanicurus X, Y, and Z. Here you see that species Y does not have the red pigment. Genetic evidence electrophoresis. DNA is cut by restriction enzymes into small segments based on the code. Those segments are separated by running them through a porous gel. DNA is negatively charged, so a current is run through the gel to separate the pieces. The smaller pieces go further down the gel. The fragments form bands on the gel. You can see a traditional electrophoresis on the right-hand side. However, for this lab, you will do a simulated gel electrophoresis. After cutting strips of paper at an appropriate point, you arrange them based on the size of the fragment to get the following result. The paper strips are the DNA sample, and you represent the restriction enzyme cutting the DNA at a specific sequence. Here you can see that species Z and Botanicurus have the exact same bands, showing, amongst other evidence, that species Z is the closest related to Botanicurus.